Hi, everyone. This is Christy Sullivan. I'm the Secretary of the American Society for Cellular and Computational Toxicology. It is great to have you all here. Welcome to today's webinar, the first ESTIV ASCCT webinar of 2021. As you can see, we've switched to Zoom. So I'll give you a brief tour of how you ask questions and such. But first of all, of course, you can see the websites of both of our organizations on your screen. All of our webinars are presented together by ESTIV and ASCCT. Both of our organizations host regular meetings and pursue other activities to promote computational and in vitro toxicology fields. Both of our organizations offer training and education and aim to support early career scientists. And this webinar is a perfect example. So first of all, just some quick housekeeping. We are recording the webinar and the recording will be posted on the ASCCT website in a few days. Also, your lines are going to be muted throughout the webinar. You can ask questions of the presenters using the question module. So you can see down at the bottom of your screen, the Q&A tab, you can just hit that and then you'll be able to enter a question. And after each presenter finishes, I'll moderate some of the questions by reading them out. If you have a technical issue or something like that, you can also use the Q&A or you can use the chat to reach us. You'll just need to send a chat to the panelists. So let's see. I wanted to briefly give some information about the next upcoming webinar. First of all, just to mention that we are starting to form the organizing committee for the 10th annual ASCCT meeting. This will be held in the fall of this year, somewhere around October-ish. So let me know if you want to join the organizing committee. And then we will have our next webinar on February 25th. And the topic is very exciting, going to be PBK modeling and quantitative in vitro to in vivo extrapolation with two presenters from Europe. You can register for that webinar right now on our website and also find a list of all the past webinars and view the recordings. And then you can also sign up on our website. Up at the top of the website, you'll see an email sign up so that you can find out when upcoming webinars and other events are happening. So I also wanted to mention that ASCCT is supporting the international workshop on QSAR and environmental health sciences. This was planned for 2020, but now is going to be in June of 2021 this year, and it's going to be virtual. You can submit your abstracts up until the end of the month, January 30th, so please do that. We hope you'll join us for that meeting. And before I introduce the presenters, I'm going to let Helena Kandarova, who is president of the European Society for Tox in Vitro, give a little 2021 introduction to some of ESTIV's activities. Go ahead, Helena.
Thank you very much, Christy, for the introduction. I will now share the screen so that you can see my slides. I hope that the view is now good. So let me welcome you to this webinar and let me wish you a little bit delay Happy New Year. And I hope that all of you are healthy and despite the complicated situation, you are more or less not affected by the current situation that is happening around the world. We had quite tough year last year. Due to the COVID, we had to change a lot of things, but altogether we managed really well. As Christy mentioned, similarly to ASCCT, also ESTIV is organizing a lot of things that aim into the promotion of the in vitro and in silico toxicology. We are devoted to the promoting the 3 R's and alternative methodologies to the scientists, and we try to strengthen the scientific network of in vitro and in silico toxicologists. STIF is organizing conferences every 2 years, and perhaps some of you know that we had to change the date of our major meeting that had to be organized in Spain last year, so we moved this meeting into 2022. But we also organize every year applied training courses. So this year, last year the training course has been organized as an online event. We also do organize workshops, like the workshops or webinars that are organized together with ASCCT, and we try to disseminate information through the number of the journals. Our affiliated journal is Toxicology in Vitro, and we are also collaborating closely with some other journals and editorial offices. This is our board that has been elected last year for the 4 years. I will not go into the details, but you can see that we have here representatives from European countries as well as United States and Japan. STIF currently counts 260 individual members from 36 countries. We have a couple of corporate members that are a big support for our society. This year in 2021, we have 2 new organizations joining us. This is the Accelerate and the Fraunhofer Institute. We also collaborate closely with In Vitro, the Italian Cell Talks, and EVTS, and signed memorandum of understanding with several organizations worldwide. So what is happening in 2021? So we are really happy to continue in collaboration with ASCCT in organization of these webinars now through the new platform Zoom. STIF will be giving several awards and also travel bursaries. One of them is the Best Poster Award at the GRC Summer School that is going to happen relatively soon. So please visit our website to get informed about the event as well as about the conditions of the Best Poster Award. Then we will also give a Best Poster Award at the Neurotox Congress and Poster Award on the World Congress 11 on the alternatives that is going to happen in autumn this year. And we are also going to provide 2 travel bursaries. Then during the Eurotox 2021, there is a STIF session submitted and accepted on the artificial intelligence and machine learning in chemical risk assessment. We have excellent speakers in this session, and I hope that we will see each other at this meeting finally live. This year, we are also going to again organize the STIF Applied Toxicology course. It will be organized together with LIST in Luxembourg. Currently, there is not yet a date set, but it will be most probably the 2nd or the 3rd October in 2021. I would like just to mention that this course is mainly for the young scientists, but also it can serve as a ERT course or the course that is aiming to the ERT recognition. And of course, we will be continuing in activities for organization of the STIF 2022 Congress in Barcelona. For the applied toxicology course, please visit the link that is given at the bottom of this page. Currently, you will find there a lot of information about the last meeting that we organized very successfully together with Bloch and UC Leuven. And if you are interested in the Congress activities in 2022, there is a whole web page set up around the Congress, and the abstract submissions already happen, but we will reopen it so that people can refresh their abstract or submit new abstract, and the call for abstracts will be reopened around November, December 2021. So that's all from me, from our end, from STIF, and I wish you a nice webinar, and I'm looking forward to these 2 excellent presentations. Thank you.
Wonderful. Thank you, Helena. Hope you all will get involved in some of those activities. All right, so now I'd like to introduce today's presenters. I'm very excited. We have two really great presenters and sharing their work today. Just a little bit of background. Each year during our annual meeting, the ASCCT gives 2 awards to students and early career scientists for their presentations during the meeting. The first award winner is Sherri Bloch from the Université de Montreal, and she was awarded the Ray Tice Tox 21 Student Award, which is presented to recognize the best student presentation during the meeting. And we were able to establish, thanks to a generous donation from Dr. Ray Tice, who has been a leader in implementing and setting the stage for toxicity testing in the 21st century. Sherri is going to be presenting using in vitro data to derive acceptable exposure levels, case studies on PBDE developmental neurotoxicity and DDE obesogenicity. Sherri has a Master of Science in Pharmacology from the University of Montreal, and she's currently doing a PhD in toxicology and risk analysis at the Department of Occupational and Environmental Health at the University of Montreal. She's also affiliated with the Center for Public Health Research in Montreal. And I'm going to briefly introduce our second presenter so that we can just move to the second presentation once we're ready. So Eva Vitucci from University of North Carolina, Chapel Hill was the winner of the Ed Carney Predictive Toxicology Award, which recognizes the first author of an outstanding poster or platform presentation that will advance predictive toxicology and is named after Dr. Ed Carney, a member and supporter of the ASCCT, who was a leader in predictive toxicology. So as I said, Eva won that award for identifying the molecular mechanisms of air pollution-induced cardiovascular disease. She's a PhD student at the University of North Carolina, Chapel Hill. She has a Bachelor's of Science in Microbiology from the University of Florida and is working on her PhD in the curriculum in toxicology and environmental medicine. Okay, so I'm going to stop sharing, and Sherri, I'll let you go ahead with your presentation.
Thank you, Christy. All right, so is it clear?
Yes, looks great. Go ahead.
All right. So hello, and thank you to the ASCCT for this opportunity to present my project. So my name is Sherri Bloch. I'm a PhD student, as Christy said earlier, under the direction of Dr. Marc-André Werner. I study toxicology and risk analysis at the School of Public Health. So today, here is my project, using in vitro data to derive acceptable exposure levels in case studies on PBDE, neurodevelopmental toxicity, and DDE obesogenicity. So here I'll be giving a brief background about my project, talk about my objective and the 2 case chemicals. I'll delve into the methodology, the results and conclusion for each of the chemicals, and some final remarks. So as we know, there are over 85,000 potentially toxic chemicals currently on the market and thousands more entering it every year. So because of this, cost and time efficient methods of producing acceptable exposure levels are necessary. However, acceptable levels of exposure are generally based on toxicity tests in animal models, which can take up a lot of time and financial resources to produce toxicity data for a single chemical, which may then poorly predict adverse health outcomes in humans. Therefore, this project proposes an alternative method of calculating acceptable exposure levels through use of available in vitro data and biological and computational modeling technologies for chemical safety assessment. Approaches using in vitro to in vivo extrapolation have been published. However, their aim was not to replace the use of animal models, but rather chemical prioritization. Another issue with current published approaches is that they have been validated through animal models. So through new methods, adopting in vitro data, ideally derived from human cells and validated against human data, are currently being advocated to replace traditional animal models. Here's my objective. My general objective is to develop and evaluate a novel approach based on in vitro data to establish acceptable exposure levels in humans. So this is just a quick view of my flowchart. This is the methodology I will be presenting. We'll be going through this chart step by step in the following slides. So it's first the literature search, then the determination of a POD, mass balance modeling, toxicokinetic modeling, uncertainty factors, we're gonna take a look at them, and acceptable exposure levels, and finally a comparison with epidemiological studies. So, here's the criteria that, so the first step is of my methodology is the literature search. Here we chose 2 case chemicals based on the following criteria. There must be a strong epidemiological evidence and association between exposure and an adverse effect, which is then supported by a meta-analysis. Then there must be human in vitro data available. Sometimes there isn't too much or there isn't any. And finally, a toxicokinetic model must already be available or feasible to make. Here are the 2 case chemicals that we chose based on the previous criteria. So first of all, we chose PBDEs, which have an adverse endpoint of neurotoxicity, and para-DDE with an adverse endpoint of obesogenicity. PBDEs are lipophilic chemicals which have a tenuous link with the products that they are meant to protect, such as furniture, textiles, and electronics. For this reason, they seep out into their environment and are found attached to dust as well as in food. They're then capable of bioaccumulation and biomagnification in the food chain. In vitro tests for neurotoxicity are performed on neuroblastoma and primary fetal neuroprogenitor cells. In epi studies, prenatal exposure to PBDEs have been associated with a negative effect on motor skills as well as behavioral and cognitive development. Para-DDE is a lipophilic metabolite, so also lipophilic, of the pesticide DDT and is assessed for obesogenicity in vitro through the use of mesenchymal stem cells and pre-adipocytes. In epi studies, prenatal exposure to para-DDE is associated with childhood obesogenicity. So the next step is the determination of a point of departure. For PBDEs, we recently received really excellent unpublished data from the Fritsch lab, where neuroprogenitor cells are exposed to BDE47, a congenitor of PBDEs, and studied for differentiation into oligodendrocytes. As you can see, there is no NOEL observed, so no observed adverse effect level. None is present, but we have a large enough data set to perform benchmark dose modeling. On the right, for para-DDE, we chose the study by Milorova and al. From 2016, which explores changes in PLIN2 gene expression in human adipocytes derived mesenchymal stem cells due to DDE exposure. So the reason why we chose this one is because in humans, a missense gene in humans for PLIN2 actually causes pretty severe obesogenicity. So again, in this one, as you can see, there wasn't any NOEL described for this study. All of them were significant. But also here we see that there's actually only 2 data points available. So it would be these last 2 here. No, these 2. This one and this one. Okay, this one. And sorry about that. And there's only 2 points, so there wasn't enough for the for a benchmark dose model. And we had to choose a LOEL, which is the lowest observed adverse effect level, as a point of departure. Sorry about the confusion. And next. So As we know, calculating the benchmark dose or the concentration allows us to identify the true NOEL, which may not be one of the concentration defined by the researcher. We chose to define our benchmark concentration as a 5% response to the chemical, and to be more conservative, we chose to use the lower bound limit of the 95% confidence interval. We calculated the nominal benchmark concentration level to be 0.00315 mole per liter for BDE 47. Of course, this is in terms of the nominal concentration, which is the concentration the cells are exposed to, not the concentration that actually enters the cell. To calculate the intracellular concentration, we need the enrichment factor, which is a ratio of the concentration of the chemical outside and inside the cell. And here at the bottom of the slide, you can see the formula used for that, for the intracellular BMDL. Okay, so on to the next slide, and this is what the mass balance model would look like. This is a representation of the mass balance model by Armitage et al. From 2014. And it's the one that we used to calculate the intracellular concentration for the chemical. Through input regarding physical properties of the chemical and the in vitro environment, it determines the chemical distribution between the headspace, the medium, and the cells. In my opinion, this is a very important step in the methodology. Every experiment imposes different conditions, such as lipid and albumin content. And every chemical reacts differently due to the complexity of BDE 47. We were unable to use it in the mass balance model and instead employed the empirical value of 60. However, we were able to use this for the model for DDE. In the experiment, we chose for DDE by Milorova and al., the medium happened to contain a very high lipid concentration, which left less free chemical available to transfer into cells. Finally, use of the mass balance model for DDE yielded an enrichment factor of 0.46. So that would mean that less than half the concentration that the cells were exposed to actually entered the cell. This is a very big difference. And use of this mass balance model was really fundamental to decode the right concentration associated with the seen adverse effect. All right, so this is the toxicokinetic model that was used. As you can see, it's a two-compartment toxicokinetic model of gestational and lactational exposure. On one side is, on the left is the maternal lipids, on the right it's the child lipids, and this is because we are under the assumption that all of the chemical will be stuck to the lipids. The chemical is then capable of transferring to the child through breastfeeding and placental diffusion. This model has been used and validated in previous studies. Here are the uncertainty factors that we chose to use in each case. So on both sides, for BDE 47 and for para DDE, we see that we chose, we didn't use any interspecies since we didn't use animals. We did need to use subchronic to chronic, inter-individual as well, so 10 for each. For in vitro to human, because we used human cells, we only did a number of 3. Only for para DDE, we had to do another one for Lowell to Noel. And at the end of all this, for BDE, we had a composite factor of 300, and for para DDE, it was 3,000. So here we're just kind of gonna recap on what we saw until now. So starting with in vitro data, which gave us a nominal concentration. We then transformed that nominal point of departure into a BMDL or the Lowell for the DDE. That number was put into the mass balance model in order to derive the real concentration inside the cell. This number, which is the human equivalent for within the child tissue. Sorry. The child concentration in the target organ. And so then after this, we use the, so in DDE, the target organ would be the adipose tissue, while in PBDE, it's the child's brain. The TK model is used in a reverse dosimetric fashion to obtain the maternal exposure necessary to achieve the target organ concentration. Certain uncertainty factors are applied at the level of maternal exposure, put back into the TK model to get a maternal concentration required for the 5% response in the cells. So do you remember previously we said the 5%, it was for the benchmark dose response. So now the question is, why is the number that we want the maternal concentration? So we went back, we put the number into the maternal exposure, we got the acceptable exposure level, we put it back into the TK model, and we find the maternal concentration in lipids. And why do we want this number? That is because this is the number that is given by the epi studies. This is what is studied. Okay. So most epi studies report associations between exposure and health effects assessed through plasma levels. So our predicted plasma levels will be compared with the distribution of plasma levels measured in epi studies. Here are the results and graphs generated by the toxicokinetic model for BDE47. On the left, we have the results in the case of no uncertainty factors. And on the right, we see the results where uncertainty factors are taken into account. For the purpose of our study, we had the mother conceive at around the age of 24 and give birth at exactly the age of 25. The maternal concentration of BDE47 is in green, and for the child, it is in blue. Before birth, the plasma concentration of BDE47 is similar between mother and child. However, at birth, a large dose is transferred to the child through breast milk. This is due to the high lipid content with the attached chemical in the milk. Without the use of uncertainty factors, the acceptable maternal plasma concentration is at 4,204 nanogram per gram lipid, while it's only at 14.02 nanogram per gram lipids with uncertainty factors. So here we've compiled available epidemiological studies detailing maternal plasma levels of BDE47. Under the table, we can see maternal concentrations without and with uncertainty factors. So it's 4,204, again, which is high above the maximum levels. But for 14.02, which is with the uncertainty factors, it's really within the range of the median and geometric means. So for our take on this particular study, firstly, that the acceptable exposure level derived from in vitro data accounting for uncertainty factors was within the range of median levels measured in epidemiological studies. Secondly, the results suggested that using in vitro data and biological modeling may actually be viable. It may be a viable alternative to animal testing to derive acceptable exposure levels. And now for the second part, for PyroPyro-DDE, these are our results and graphs generated by the toxicokinetic model for PyroPyro-DDE. Again, on the left, we have the results without uncertainty factors, and on the right with uncertainty factors. As before, we had the mother conceive at around the age of 24 and give birth at exactly age 25. This model, in this model, the maternal and child plasma concentration of PyroPyro-DDE are equal before birth. But once the child is born, it receives again a hefty dose of PyroPyro-DDE during breastfeeding. Here, the calculated maternal plasma concentration at birth is 42.15 nanogram per gram lipids. Without uncertainty factors, 0.14 nanogram per gram lipids. Again, these numbers represent acceptable levels at 5% response. So in this table, you see the compiled results of several epidemiological studies for para DDE found in maternal plasma at birth. You will notice that both with and without these uncertainty factors are calculated maternal concentrations are far below the means and the geometric means and the median. So from this study, we were able to conclude firstly that acceptable plasma levels derived in vitro data was derived using the in vitro data was well below the range of median levels measured in epidemiological studies, even without the use of uncertainty factors. Second, the use of a point of departure based on gene expression, which is it's a very early response, along with a high composite uncertainty factor may be too conservative, as the predicted acceptable maternal concentration was far below the medians and geometric means found in people. Overall, here are the impacts and our conclusions. Our final reflections on this methodology is, to our knowledge, it is unprecedented for an in vitro to in vivo approach to be evaluated against human data. This methodology is cost and time effective. Here we were able to generate reference values in relation to adverse health effects. Moreover, we were able to overcome having to use an uncertainty factor for interspecific extrapolation, since we used human cells. However, there is still an obstacle to be overcome, and that would be uncertainty factors. To our knowledge, there is no precedent in the application of uncertainty factors when extrapolating from human in vitro data to human in vivo data. We're still trying to overcome the kinks in that respect. So, and that's all for my my presentation. I'd like to thank my advisor, Marc-André Werner, for his continued help and support, as well as all my lab mates and their continued aid. Furthermore, a big thanks to our collaborators in the Fritsch Lab who provided us with excellent data and answered all of our questions. Another huge thanks to James Armitage and Inku Kramer for their guidance in mass balance modeling. And thanks to Maryse Bouchard and Chandru Chandrasekera for their support. This project was funded by NSERC, the FRSQ, and the Canada Research Coordinating Committee. Thank you. Any questions?
Thank you, Sherri. That was a great presentation.
Thank you.
For added a little bit too from your ASCCT work, so that's great. Thanks. I don't see any questions yet in the Q&A box, so let us know if just go ahead and type in your questions. We can also have typed, Q&A with Sherri while Eva is presenting. I have a question for you, though, which is, I think this question of trying to link in vitro outcomes to in vivo exposures and epidemiology I'm really glad you're doing this work, because I think it's really important to try to link these two pieces of human data. And I wonder if you've considered or thought about the adverse outcome pathway framework as a way to kind of provide a link between, the early events in a pathway and the epidemiological information.
Yes, I've read about it and I've looked into it, but right now we're a little bit more in the earlier stages, just trying to validate this kind of methodology and show that it can provide some kind of data.
Mm-hmm.
Yeah.
That makes sense. Okay, one question coming in is, have you published this work yet, or is it?
Not yet, not yet. It's still something that I'm writing it. I should be it should be published sometime this year or early next year. Just takes time, I guess, until the revision. And there's also another paper I have to work on before.
Yeah.
Yeah.
Another question is, it's often there's no way to link human health effects to a specific chemical. And so I think they're referring to sort of findings in epidemiology. And were in the epi studies you examined, were the health effects known specific to these chemicals?
So that's really why we start looking at the meta-analysis. So when you bring in all of the in vitro studies and put it together with the epi studies, altogether you're able to see that there is really a link. And so that's why one of the criteria for choosing a chemical is to have meta-analysis available.
Great. And also I was wondering, just thinking about, you said you're planning to continue working on uncertainty factors. This is also a lot of interest, because they're used quite a lot in regulatory risk assessment, and often it seems they're sort of applied because we're not really sure, so we just add an uncertainty factor. And I'm just curious about whether you have some ideas about what you're thinking in terms of trying to add some more bounds around, or some more data to be able to refine the uncertainty factors.
I really can't say just yet. It's really unprecedented right now, jumping from human in vitro cells to humans, and I really don't know what my next steps are, but I'm really planning on doing a lot of research to support my my methodology concerning the uncertainty factors.
Great. Another question has come in asking about exposure to mixtures of chemicals. Have you thought about that?
Yes, I've thought about it, but we're really in the earlier levels, and so we're really just trying to do one chemical at a time right now. But it's definitely important, the effects of chemicals mixed together, whether it could have an interaction and cause a different kind of effect altogether. Yeah, it's something that I guess will be worked on in the future.
Okay. All right. There's a couple of questions coming in, but I think we'll leave them to see if you can answer them in writing, because I think we're going to move on to the next presentation. So if you could stop sharing, and then we can let Eva present. Eva, go ahead and share your screen.
Okay. All right, how is it looking? Can you guys see the main presentation and not my notes?
Yes, looks good.
Perfect. Okay, great. Well, thank you for the introduction earlier. That was great. And so I'm excited to talk to you all about my research today. So again, we're going to focus on kind of the molecular mechanisms of how air pollution can induce cardiovascular disease. But before we get into it, because this research is funded by the EPA, I just have to say that what you're hearing today is a reflection of my views and my findings, and does not necessarily reflect the views and the opinions of the EPA. Fun disclaimers. So as we all know, new chemicals, metals, and toxicants are entering our environment on the daily, and they'll continue to do so for years to come. And unfortunately, this rate of production vastly outperforms our ability to regulate these agents and assess the impacts that they have on human health. And so while animal studies have historically been employed to help us identify the effects that these agents may have on human health, these studies are limited by time, sample size, cost, etc. And in addition to these limitations, animal studies often focus on physiological endpoints that oftentimes leave the molecular mechanisms driving those endpoints still unclear. And so as a result, the field of toxicology, as you all may know, has begun emphasizing this idea to switch to a high throughput and computational toxicological approaches to help us elucidate those effects of that ever-growing list of agents that humans are being regularly exposed to and the effects that they have on human health. Agents that we're regularly exposed to false particulate matter. And so particulate is just the collection of all of the solid and liquid particles in the air that we breathe. It arises from multiple different sources, as you can kind of see in this infographic here. The main source is vehicular emissions, but it also can arise there from agricultural practices, dust, smokestacks from industries. Particulate matter is that it's a strong pro-oxidant, so it oftentimes causes redox dysfunction when it comes in contact with cells. It can be mutagenic, but the most thing that begun to identify is that particulate matter is tightly linked with cardiovascular disease. So large population studies, hospital records, and animal studies all provide evidence that our cardiovascular system is negatively impacted by breathing in air pollution, and specifically particulate matter. And this can occur in populations, whether they're exposed to particulate matter regularly or acutely, so just a few days at a time. And unfortunately, this results in instances of stroke, heart disease, as well as respiratory diseases that oftentimes result in mortalities. And so the World Health Organization estimates that air pollution kills around 7 million people worldwide every year. How exactly particulate matter causes those diseases and mortalities is still relatively unclear. Seems to be a main driver of the negative effects of air pollution. And there are a couple of main mechanisms of how this can happen, but the molecular side of that is still largely unclear. But just to kind of bring us all up to speed, what we believe is happening is the lungs, and they reach the lower levels of the lungs as well. Once they're there, they can generate lung inflammation, which then secretes inflammatory mediators that can then leave the lung, enter our cardiovascular system, and cause cardiovascular impairment. This is kind of thought as the indirect mechanism. The second main mechanism that's proposed is the direct mechanism. So that is that the enter lungs, and they're small enough to basically leave our lung and directly translocate into our bloodstream and our cardiovascular system. And then again, once they're there, they cause vascular impairment. Now, these impairments come in a variety of different colors, but they oftentimes show as impaired endothelial functioning. So that is the cell type that lines our blood vessels, which can oftentimes lead to increased vasoconstriction, as well as improper ability to dilate when needed to. And then these impairments manifest as different cardiovascular diseases that I've mentioned before: heart attacks, strokes, and atherosclerosis. And so while the main take is that while we know that cardiovascular disease oftentimes is to be a key middle step, is that our vasculature, so our blood vessels, appear to be the first target that gets
Eva, I'm so sorry to interrupt, but it seems like you're cutting out a little bit and there might be a problem with your internet. So I wonder if you would pause your video, that might help.
Yeah, let me pause. I can stop sharing and I'll stop video.
I think you can go ahead and share your slides. The video should clear it up.
Are you guys looking at the right screen again? Uh-oh. Hello?
It was up and then it went down.
Okay, hang on. Let me try one more time. Sorry.
No worries.
Okay, how are we looking?
That looks great.
All right, so should I just keep my video off then?
Yeah, let's try it like that.
All right. But okay, so clearly what we've identified is that our endothelial cells and our vasculature appears to be a main of particulate matter exposure and mediating all those cardiovascular diseases. So to take those studies one step further and to help identify what those molecular mechanisms are in our vasculature, I decided to develop an in vitro model to identify the effects that may be a model that replicated the alveolar capillary region, which is the model of our lungs, or the location in the lungs where particulate matter oftentimes can interact with our alveolar. Again, particulate matter can be so small, it can reach those distal lung environments. And so this environment is quite complex, but I'm just going to show you all a simplified version of it today. It consists of an alveolar epithelium that obviously lines that alveolar air sac. It consists of alveolar interstitium, which are fibroblasts that help keep that alveolar air sac open and supported. And then it also consists of capillary endothelium, so that capillary blood vessel network that surrounds your air sac and is really important for facilitating the movement of oxygen and carbon dioxide into your bloodstream and out of your bloodstream when you breathe. And so I had two main goals when I wanted to develop the model of this region. The first goal was that I wanted to make sure that the model reflected the architecture of this region as much as possible. And then the second goal was that the model would reflect the functionality of this region. And so our goal was to take advantage of transient inserts and to use commonly available cell lines to reflect the cell architecture. And so I used alveolar epithelial cells, or alveolar-like epithelial cells, the H441 cell line. And I was able to seed them on the inside of a transwell insert. IMR90, or the fibroblast cell line. I was able to seed those on the opposite side of that insert. And then I was able to use the Hulex cell line, or the primary lung microvascular endothelial cell line, to seed at the bottom of that transwell dish. And when the cells are seeded in this manner, they not only reflect the cellular architecture of the in vivo acre region, but they also allow you to investigate the effect that a direct exposure of your alveolar epithelial cells have. And then you also are able to investigate the indirect exposure that that stimulant may have on those underlying endothelial cells. So a huge benefit of this model is the ability to separate by cell type and understand how each cell type responds differently to your different exposures. And so another second goal of ours, as I mentioned before, was that this model reflected the in vivo functionality. And so one of those main functions of this region is to form a tight barrier that's relatively impermeable. And this is essential to ensure that the alveolar air sacs remain open and dry for that efficient gas exchange. And so the epithelial cells in this region, those alveolar epithelial cells, are largely the main cell type that can promote this from happening. And so I wanted to investigate if the epithelial cells in our model also formed impermeable barriers that had tight junctional proteins present. And so to do this, I performed 3 assays. I performed immunofluorescent staining for the presence of tight junctional proteins. And as we can see, we had great success in identifying the tight junction protein Z01 in our epithelial cells. It's migrated perfectly or nicely to the cell membranes, where it then binds with other cells and creates that tight junctional impermeable barrier. And we also identified great staining of the adherence junction protein E-cadherin. In addition to the image of tight junctional proteins, we also identified a function of the, we had performed transepithelial electrical resistance assay, or TEER, which allows you to assess the ability of small ions that are able to pass through. And so essentially what you see is that in the presence of vehicle alone, which is just media alone, and diesel, that the addition of diesel exhaust particulate matter, which is our particulate matter model, does not disrupt the barrier formation of the TEER. So low values on the y-axis here suggest low movement of those ions. And then using a Fitzy-Tritzy assay, which again is another permeability assay, we were also able to assess that compared to no cells alone on that insert, that when you add in your vehicle or your diesel treatment, that again, there is very low migration of the small molecules getting into that basolateral compartment. And so together, these studies suggest that we have an epithelial layer that forms an impermeable barrier that is lined by tight junctional proteins. And so we are super happy that the barrier does seem to reflect, or the model seems to reflect, the barrier integrity of the in vivo scenario. And then I just wanted to quickly note 2 or a few other significant impacts that this model has, which is that it's medium throughput, so it's not necessarily high throughput, but we've been able to get it down to a 24-well format. That we used, as I mentioned earlier, particulate matter, but you can kind of switch any particulate matter you want or other stimulant, and that you can also substitute these cells with primary cells. But to get back to our main focus, knowing that this model reflected the in vivo architecture and function, we then wanted to test our hypothesis, which was to see how TA depth exposure, that trans-alveolar depth exposure, induces endothelial redox dysfunction, and if it causes endothelial dysfunction in general. So I guess I'll go back, but our first goal was to expose these epithelial, which was our model particulate matter. And then in the endothelial cells. And so our first assessment was to investigate how the antioxidants of our endothelial cells were affected by this indirect exposure. And so we just did qPCR and Western blot analysis to assess 2 canonical antioxidant genes, heme oxygenase 1 and NADPH quinone oxidase 1, or NQO1. And what you can see is that over time, there is a great expression, induced induction of HO1. This largely occurs at 6 hours, and then the same thing with NQO1, which largely occurs at 24 hours. And again, this is all in the endothelial cell line, the Helix cell line, but we've also repeated this with primary endothelial cells to assess if there are any differences. And it seems to have a similar pattern. And then to assess if these gene expression changes were reflected at the protein level, again, we performed Western blot analysis and determined that not only were these antioxidants being expressed, but they were being translated into proteins, suggesting that the cell needs these antioxidants present to handle some sort of redox stress. And then another thing we wanted to investigate was the transcription factor NRF2 to understand if that have a role in transcribing these antioxidants, but also because we know that NRF2 is frequently activated in the presence of oxidative stress. Again, that NRF2 was activated in response to this indirect exposure. So endothelial cells are undergoing some kind of redox stress after the epithelial cells are exposed to diesel exhaust particles. But we know that the expression of antioxidants alone is not necessarily enough to claim that endothelial redox dysfunction is happening, because it could be that the antioxidants are able to raise up to the challenge and handle the situation on its own. So to investigate if other elements of the cell's antioxidant system were perturbed, we wanted to investigate the cell's glutathione antioxidant. This is again way more complicated than the diagram I'm showing you here, but a simplified a plethora of these glutathione molecules that in the presence of reactive oxygen species can facilitate the breakdown of those reactive oxygen species into water. And then that reduced glutathione becomes oxidized. Now, in order for this cycle to continue and to be able to continue fighting reactive oxygen species, this oxidized glutathione must be reduced by the glutathione reductase, where it can then go back to its starting position and start the cycle over. So of this system is key for a cell to be able to maintain homeostasis. But when this system is disrupted, what you often see is an acute here, and you often see the cell going into redox dysfunction. And so one way to assess if this is happening in our endothelial advantage of this ROGFP molecule that is reduced and oxidized. And when it's reduced and oxidized, it expresses different excitation maximums. And so basically you can think of it as different colors. So when it is reduced by the same cells, the same enzymes in the cell that reduce your glutathione, it has a higher expression at 48, so the green channel. And when it is oxidized by the same enzymes in the cell that oxidize your glutathione, it is kind of expresses at in the 4 or 5 channel, so the blue color. And so what you can do is you can put this ROGFP molecule into your cells and have an idea of if you have more of the green color or if you have more of the blue color. So do you have more oxidized accumulation or more reduced? And so what we transduced our endothelial cells. We performed our TA-DAP experiment over 3 hours. And what we found was that of more of the oxidized glutathione in our cells than the reduced glutathione. So this is the ratio here. Was that using a cell ROX assay, we also had an accumulation of reactive oxygen species over 24 hours. So we had the doubling of reactive oxygen species in our vehicle, and then over time, we had about 1.5-fold increase. And so collectively, this data suggests that we are undergoing, or the endothelial cells are undergoing and that the glutathione antioxidant system is not able to handle it, suggesting that we are undergoing endothelial redox dysfunction. And then we wanted to just investigate the glutathione system one step further. And what we observed was at the qPCR level, the different components of that system are not necessarily increasing as they should. So over time, we have an increase GCLC, which are the two main substituents that make that reduced glutathione molecule. But what we assume and what we're understanding now is that our glutathione reductase, so that molecule that is really important in reducing that oxidized glutathione back to its original state, is not being induced as much as it should be. And that's really reflective here at the protein level too, where everything is basically hovering around 1. So potentially if the cell was able to increase its glutathione reductase, the cell would be able to handle the stress of the reactive oxygen species much better. And so how does this all kind of tie into cardiovascular disease? Well, we know that redox dysfunction is tightly linked with causing plenty of downstream negative effects. And so prothrombosis is actually one of those downstream effects. So we wanted to investigate if that was happening in our endothelial cells, as we know that's a common frequency with stroke and heart attacks is the formation of blood clots in the blood vessels. So what we identified was that our endothelial cells are also undergoing a prothrombotic procoagulant gene expression profile. So they of the tissue factor protein, or F3 here, which is the main prothrombotic gene and target. And then the cells, the endothelial cells, are actually having a slight decrease in PLAT, PLAU, and THBD, which are the main genes and proteins that are essential for preventing the formation of fibrin clots and blood clots. So essentially what we're seeing is a shift to a prothrombotic, pro-fibrin clot nature in the endothelial cells when they undergo endothelial redox dysfunction. So to conclude, essentially we've developed an in vitro acre model, or the alveolar capillary region model, that is able to reflect the in vivo barrier of that region. The acre model can be used to identify mechanisms of cardiopulmonary mutagenesis, and can, the TA-DEP exposure that we've done has also revealed that endothelial redox dysfunction is occurring and that a prothrombotic gene expression phenotype may be happening. And so I think this model has really helped us understand how different cells in the body respond to different stimulants and respond differently. And it has a lot of application and ability to be used to study different stimulants as well. And so I just want to end here and say thank you to my lab and all the support, my various funding on the left. And if we have any questions, I can take them now.
Thank you, Eva, very much for that presentation. We have a couple of questions already coming in. Again, if you have any questions for Eva, you can type them into the Q&A box. The first question is, have you used this system for testing of any component of particulate matter like PAHs? I assume that means polyaromatic hydrocarbons.
We have kind of parsed out exactly what of the particulate matter can be driving these effects. I've just evaluated gene expression of different enzymes that may break down different constituents. So for example, polyaromatic hydrocarbons, I've evaluated the expression of CYPs, CYPS1A and CYPS1B1, and both of those are increased. So it could be the polyaromatic hydrocarbons have a good role in driving the effects that we see, but we haven't done any sort of isolated exposure to really confirm that yet.
Okay. And another question is your cellular model available commercially, or is it Only for internal use at this time.
It is not available commercially. I'm not exactly sure how things like that work with the EPA and if we could sell it. I don't think that's a thing, but I'm definitely open to sharing how to build it in your lab, because it's definitely approachable. That was one of the main reasons why we developed it in this manner, was so that other labs can copy it to study their stimulants of interest or plug in their favorite cell type.
Great. And another question about, well, I think they're asking two questions here actually, but the question is about how you've decided which exposure levels to use. And then they mentioned that the exposure for air pollutants can be chronic. So I think that's exposure levels, and also, have you thought about multiple exposures or chronic exposures?
Great, two great questions. The first, so answering that one is tricky. The field has a couple of papers published on, the dosimetry and the idea of how much of the particles land in your alveolar compartment and how much you're exposed to it at one time. And so we based our dosing, which is 100 micrograms per 20 centimeters squared, based off of papers that we found that have estimated that is a relevant dose that humans can be exposed to. It's a high dose that humans would be exposed to on the daily, but it is the closest we've gotten to finding data that supports that. And then also one thing, so the chronic exposure is very interesting. In the alveolar compartment, your alveolar macrophages are the main cell type that can kind of remove the particles. So in the upper airway, you have your mucociliary transport. That's really important for getting stuff up and out of your lungs. But in the lower compartment, you don't have that. And so it's found that these particles can oftentimes reside in your alveolar compartment and just there and accumulate and elicit their effects, because oftentimes the alveolar macrophages can get overwhelmed and aren't able to remove all of the particles that are needed to be removed. And is tricky because, again, that makes it harder to estimate how much is down there at any given time. And then as far as doing a repeated exposure in vitro for chronic exposures, challenges. We haven't tried to tackle that just yet, as I'm trying to kind of wrap up and finish this work so that I can graduate. But it's a great question. And I know that other people in the lab are interested in trying to optimize an in vitro model so that they can perform chronic exposure.
Yeah, and I think that question of clearance is a tricky one.
Yeah, it is very tricky for this region of the body, for sure.
So there's just one last question, and is this work published yet?
It is not. Working on it, it'll be published hopefully by the end of the semester. Well, it will be published by the end of the semester. Yeah, we're still working on some mitigation strategies, and we have some targets that we think are driving the effects that we're gonna play around with. That way we can actually publish with identified drivers.
Super.
Once we have that, it'll be good to go.
Super. Well, stay tuned, everybody, for more from both of these scientists. And actually, I'm just going to highlight too that Dr. Mokala just sent some information, and I popped it into the chat about how to get in touch with Eva and Sean in case you want more information about the model. So thanks again, Eva. Thanks, Sherri, Bloch, as well, for your presentations, for your work. And we really appreciate all of you for listening. And we will see you in February. Have a great afternoon, everyone.